Today we are presenting a method of detecting central nervous infection by an alpha virus within a murine species using bioluminescent technology. This method utilizes TC83, the live attenuated vaccine strain of Venezuelan equine encephalitis, with an inserted firefly luciferase gene which we can use to track the replication of the virus as it spreads within the host. In a recently published vaccine paper, we demonstrated our capability to visualize the invasion of the CNS and use it as a study endpoint for testing the efficacy of vaccines and antiviral treatments before clinical disease manifested in our controls.
Hello, my name is Allison Poussard, and I'm an animal technologist in the high containment labs here at the University of Texas Medical Branch. The work that you'll see here in this video was completed by me, Michael Patterson, who's a graduate student here at UTMB, and Katie Taylor, a senior graduate student in our lab. All of the work that you'll see was completed here in the Galveston National Laboratory with full IACUC approval following environmental health and safety regulations.
Utilizing in vivo imaging systems, IVIS, allows us to reduce the number of animals required within our studies due to our capability to generate images on a daily basis for each individual animal. An overview today will be showing the preparation of the animal, including forehead shaving, BMDS chipping, intranasal infection with TC83, intraperitoneal injection of luciferin for imaging, the handling and transfer of the animal from the biocontainment cabinet into the imaging box. From the imaging box, we'll be transferring the animal into the calipers imaging unit, the actual imaging process,